this is a recorded video for week 2, day 1. Today we have quite a lot of announcements, but I want to take some time to go over each of the announcements with you. So first of all, for Farm Century, we do have the presentation from them last week and also the week before fall break. So we really want to take a time to write a thank you card from Miss Ariana coming from all the way to New York City to join us for this virtual presentation. And there's a lot of program out there that will charge us hundreds of dollars just to present to us, but they are doing this completely for free. So we just really want to show our gratitude to them. So if you have a moment, go ahead and go to your Google Classroom and click on classwork and go right here to optional thank you card for Farm Century. When you click on that, it will show this template. Thank you, Miss Ariana. You can change colors, fonts, add picture, and just write down one thing that you have learned. And then we'll compile all of this together at the end of the week and send it to her. Falcon News volunteer, we're also taking volunteers who would like to share what they have learned on the Falcon News. So again, I asked a few times in the classroom so far to see who is interested. But if you're still interested, please do let me know and email me. Next one is the Spirit Week. So this week is, besides Halloween, it's very exciting because we have Spirit Week as well. So there's different theme every day. I'm going to go over what theme we have each day and how you can earn extra credit. So again, you can find that in our Google Classroom under classwork, week 2, extra credit Spirit Week. Let's click on this. So there is different theme every day. Monday is our Falcon Monday, so you can wear green to show your Falcon pride. Tuesday, you can wear anything related to Disney or Marvel. We will also have a trivia during lunchtime, so you can log into the meeting code BobX70 to join our trivia. Wednesday will be an asynchronous Wednesday. On this Wednesday, you only attend your first period and the rest of the day is going to be independent work time. It will also be a pajama day, so you can wear school appropriate comfy pajama for your first period. Thursday will be an 80s day, so wear bright neon color and side ponytail to make the 80s. And Friday, of course, you will get to wear your Halloween costume. Please to make sure that they're school appropriate, and you can also attend the costume contest at BobX17 during lunchtime. So with that said, it really depends on which day you have my class. So for our period, you have my class on Monday and Thursday. So those are the 2 days that you want to dress up for. For my even period, I'll be seeing you on Tuesday and Friday. So those are the 2 days that you want to dress up for. Okay, so you get 2 points for each of those days. And again, just like we mentioned earlier, this Wednesday will be an independent work day, so you will only meet with your first period. Last but not least, extra credit. So if you attended the Bay Area Science Festival last week, make sure to turn this in sometime this week. It is due at the end of the week. Now, if you are still interested but you did not get a chance to join the Bay Area Science Festival, I also posted a few videos based on what we have learned so far and what we will learn in the future. So let me show you how that looks like. So again, week 1 right here, extra credit Bay Area Science Festival. When you click on this, you will see 5 video links that I posted. It is Life Science with Chabot Space and Science Center. They talk about the scientific method as well as they talk a lot about observation. So you can watch that. There is also Can You Believe Your Eyes? tour to brain lab, and this one they take you to an actual science lab and they show you the human brain versus mouse brain, and really talks a lot about what we'll get to in the next section in body system. So if you're interested in brain and body system, feel free to watch this one. I also include one about wildfire because we have so many wildfire in California in this past year, and you can also learn more about in this video. And last but not least, if you can speak Spanish, we also have a video that is only in Spanish and is talking about career in science. So you can also click on that. So again, if you are still interested in the extra credit, make sure to fill out this form. This includes a screenshot and also a paragraph, 3 to 5 sentences on what you have learned so far. With that, let's move on to our next part. If you have any question about the announcement, again, make sure to email me and let me know what your question is. Now for that, we are going to go over week 1, day 3 virtual lab. Now this is an assignment that we started last week. Depending on which class you are in, some class we didn't have as much time to cover this lab, so we want to go over it this time. So again, you put your name right here, period. Question 1. I am a protective lipid bilayer surrounding each cell. I regulate what enters and leaves the cell. What am I? And that is the cell membrane. The cell membrane regulates what molecule go into the cell and what molecule go out of the cell. For example, water molecule or sodium, chloride. So how many molecules can go in and how many molecules can go out? Question 2. I am a jelly-like substance composed mostly of water that lies between the nucleus and cell membrane. What am I? This one would be the cytoplasm. So the cytoplasm is the fluid within the cell and it fills the entire cells. Question 3. I am the cell's DNA. What am I? This one right here would be the nucleus. Nucleus contains a genetic material called DNA within the cell. It is also the control center of the cell. Question 4. I am a network of sacs that are responsible for manufacturing, processing, and transporting chemical compounds used by the cell. What am I? This is the endoplasmic reticulum that is around the cell. On the endoplasmic reticulum, there are also little dots called ribosomes that are responsible for making proteins. And they make proteins based on the instruction given by the nucleus. So right here, question 4, we have endoplasmic reticulum. Question 5. I am a stack of membrane that collects, modifies, and sends chemical compounds to other cells' organelle. What am I? So right here, this will be the Golgi apparatus. So after they make proteins in endoplasmic reticulum based on the instruction from the nucleus, they send these proteins that are made to Golgi apparatus. Golgi apparatus is like a post office, UPS or FedEx. What they're responsible for is to say, well, this protein go to the lysosome, or this protein go to the cell membrane, this go to the nucleus. So they send it to different location. So that is Golgi apparatus. So number 6, I am the site for protein synthesis in the cells. You can find me in rough endoplasmic reticulum and throughout cytoplasm. What am I? So this dots right here, we can see in the cytoplasm, in the fluid within the cell. We can see a lot of them on the endoplasmic reticulum as well. They are in charge of making proteins. So those are called ribosomes. Question 7. I am the powerhouse of the cell that convert nutrient into energy that can be used by cells. What am I? That would be the mitochondria. or for singular form, mitochondrion. So mitochondria, what they do is that they take sugar from the food that you eat as well as oxygen. Together, they are going to generate energy. In seventh grade, you are going to get deeper into understanding how mitochondria work in a process called cellular respiration. But for now, just know that mitochondria, they convert nutrient into energy. Question 8. I break down all and non-functioning organelles with enzyme and chemical. What am I? That would be the lysosomes. So lysosomes, again, they are like the trash can and the recycle bin in the cells. They are responsible for breaking down all organelles. Question 9 and 10 is an organelle that only plant cells have. So in number 9 it said, I am a porous outermost layer that surrounds that plasma membrane. I protect and support the cell. You won't find me in an animal cells, but you can find me in a plant cells. What am I? So this one right here is the solid structure around the cells. So of course, both animal and plant cells, they both have cell membrane. But what is different about plant cells is that in additional to having a cell membrane around the cells that regulate what goes in and out, there's also another layer called cell wall. This is a solid layer, a rigid layer around the cell. So this is also a reason why you see that plant cells, they are more like a polygon shape or square, rectangle, or even hexagon. Is because of this structure that they have around it called cell wall. Versus on the other hand, you can see that animal cells, they are more circular and they're more round. Last but not least, question 10. I convert sun's energy into chemical energy through photosynthesis. I'm the reason why plant cells are autotroph. What am I? So this is the part that is called chloroplast. This is the reason why plants are green. This is also the reason that why plants, they do not need to eat food. They have the ability to make their own food with carbon dioxide, sunlight, and water combining together. And after they make their own food, those food can go into mitochondria to make energy. So again, this will be chloroplast. Again, if you did not get a chance to complete this assignment yet, please complete this assignment by next week for 70% credit. With that said, we are going to go into an independent work right now, which is the SEP simulation. Let me show you how to complete the SEP simulation. The SEP simulation is going to be in your STEM scope and the Google form. Again, it will be in your Google Classroom. Let's take a look at the SEP simulation. To go to STEM scope, make sure to click the waffle icon. Go all the way down to STEM scope right here. After logging to STEM scope, again, you want to click on the SEP simulation. I'm going to log into the student account to show you how that looks like. So right here for period 1, you can see right here it says SEP simulation for the cells. You also see this note that say make sure to fill out the Google form here, and it has a link. Let's click on this. I'm going to use full screen for this section. Right here in this lab, you are the forensic investigator working for an international law enforcement agency.
Please enter your first and last name. Some organs used for transplant have been found to be diseased. The hospitals need your help discovering which organs are diseased so that they can be disposed of.
After each section, make sure to click continue.
Your job is to conduct an investigation in order to discover which organs are diseased. You have been given hundreds of organ biopsies to analyze back in your lab. However, you can only analyze so many at a time. Therefore, you are focusing on only 9 slides today. First, you must identify which organ the biopsies belong to. You may refer to your reference guide in order to identify the tissue samples.
Now on the left right here, you see 9 slide from 102A all the way to 102I. And right here, you also see the picture of different biopsy. So you can see a healthy heart, a diseased heart, healthy liver, and a diseased liver, healthy kidney, and a diseased kidney. When you click on the Google form, you also see a very similar picture that I put together. So there are 3 organs basically: heart, liver, kidney, and for each organ you will see the healthy version and also the disease biopsy.
Let's begin with slide 102A. Drag and drop slide 102A into the microscope to begin your investigation. Identify which organ this biopsy belongs to by clicking on the name of the organ whose cells visually match the cells in the biopsy. This data will be recorded in your pathology notebook.
So right here, take a look at this sample. Which one does it look similar to? You might notice that it looks very similar to this picture of a healthy heart. So in notes, I'm going to click on hearts.
Now you will identify if the organ is diseased by seeing if the biopsy visually matches a biopsy of a diseased organ. Refer to your pathology reference guide for help.
So again, earlier we can see that it's a healthy heart, so I'm going to put that it's not diseased.
Based on your research, do you believe this organ to be diseased? Now you must check your work. Drag and drop a dropper from the vial of trichrome stain onto the slide. The vial has a chemical that will activate if it comes in contact with certain proteins that are known to be present in the disease. This information will be recorded in your pathology notebook. It looks like slide 102A is not diseased.
So right here, we can tell that this is a heart organ and this is not a disease, it's a healthy heart. Now you have 2 choices. You can complete the whole lab and complete all the data first before you record it in your Google form. So you can complete all of them before recording it here. Or you can do one at a time where each one you complete and then you record it in your Google form. Either way is completely up to you. Now, just a reminder, in this lab, they only check whether you get the disease or not correct. They don't check whether you get the organ correct. So make sure to be very careful with that. But of course, in your Google form, it is auto-graded, so then it will check for both the organ and whether it's healthy or not. Okay? So I'm going to show you 102B again as an example.
Now analyze slide 102B. Identify which organ the tissue sample is from.
So again, you drag the slide to the microscope. We identify which one it is based on the picture. So this really requires observation skill. But you might see that these 2 pictures look very similar, and this is a diseased liver. So I'm going to put liver right here.
Based on your research, do you believe this organ to be diseased?
So I'm going to put yes based on the picture that I see.
Check your work with the trichrome stain. It looks like the amyloid protein is present in the tissue. This means that the organ for slide 102B is diseased.
So right here, again, I said diseased liver. Now, if you click on the wrong one, they're going to have you click on the right one before they let you continue. So please be careful when you complete the lab on Wednesday. So right here, we're going to go to 102B and I'm going to put a diseased liver. So that's how you're going to complete slide 102A all this way to slide 102I. Okay, you do want to make sure that you have 6 or more correct when you complete this assignment. And on the very first question, we also have one additional question for you to make it 10 questions altogether. And he's asking you to choose one of the tissues of your choice, any of the 6 right here. Healthy heart tissues, diseased heart tissues, healthy liver, diseased liver, healthy kidney, and diseased kidney. Choose just one of the 6 and describe how it looks like. Okay, you can describe the colors, describe what kind of shape do you see, so you can use your observation to do that. All right, so with that, I do want to do a preview for this Wednesday. So this Wednesday is a catch-up day. So for the class who didn't have as much time for your independent work, so you are going to spend most of your time to complete week 2 day 1 SEP simulation that we just went through. For the class that have independent time, you do want to double check that all your work so far they are complete. So week 1 day 1 that we completed last Monday, week 1 day 3 virtual lab that we went over today, week 2 day 1 SEP simulation, and also to make sure that you finish up to slide 11 in your notebook. And of course, if you finish, you can also work on the extra credit for Bay Area Science Festival or to fill out the thank you card for Miss Ariana for Farm Century. Have a great day and I'll see you on day 3. Bye bye.